that was a very enthusiastic title when I got asked to, and I'm going to speak to you about what my lab is doing. 5 more minutes before coffee. Yes, so my lab is interested basically in understanding what is generating brain complexity. And I'm going to try to, oh, maybe not. Yeah, that's what I'm trying. Doesn't work. Anyway, generating brain complexity in 2 ways, and this during development. And when I say development, I mean before birth. At one level, the tissue level, we look at very early events, which I'm not going to speak about, that are important and that we found critical to define the future size of your forebrain. And you can basically switch temporarily between a small to a big forebrain by actually tuning the time at which you induce certain signals inside the forming brain. Now, what I will speak about is the second side of my lab, which actually look at generation of complexity at the level of the neuron. And so in the neuron, we know that the neuron is a very big beast, a very special cell, because compared to any other cell, it's actually incredibly big. The example on the right is showing you a typical corticospinal neuron that basically have the cell sitting in your cortex up here, and local action is actually happening centimeters to sometimes meter away, if you're tall, from the cell body. This is an issue because it actually raises all sorts of questions about how you can actually make local decision when you have such a distance between the local decision and the nucleus. Not only that, when one looks at what's happening in the evolution from fish to human, one sees that instead of going from one gene, one transcript, one protein, we get complexity in transcriptomics such that you go to human with one gene, many transcripts, and many proteins. And the example there is neurexin, where in human, neurexins can go from one gene to above 100 different transcripts. They do that by cutting the draft transcript into different versions, and this cutting mechanism is called splicing. And it's requiring proteins called splicing factor and spliceosome proteins. Now, this, and unfortunately I don't have a pointer, but this is happening in the nucleus down there, where the draft RNA is sliced and prepared into different versions. Now, as this is happening, then the transcript is traveling outside of the nucleus and into different parts of the axons and dendrite, and this is a mutation from MAC to PC. So you can see that they're outside, but they don't go out of the neuron, they actually stay in. Anyway, so the point being that they travel, and there are 3 things that we don't understand. We don't understand how they're transported, we don't understand how they stop at the right location, and we do not understand how they decide, or something else is deciding to translate them into proteins at the right time at the right place. I have no answer to any of these questions, but we are walking towards them. And so the point is that the weird thing that has been discovered is that, from our lab and others, is that these transcripts are not transported out of the nucleus all naked, but they are actually transported with so-called RNA binding proteins, but some of them being splicing factors and spliceosome proteins. And these are not supposed to be anything else than proteins that are cutting the transcript in the nucleus. And so my lab has been really passionate now about trying to figure out how these splicing factors work outside of the nucleus. We know by genetic tricks in animal models that if we actually prevent these splicing factors to be active outside in the axon, we have tremendous defects. And so one is we have very abnormal axons, but the other is also we have no synapse formation. So clearly, outside of the nucleus, the spliceosome protein splicing factors are required. And what we discovered too is that the mechanism by which they act is very complex. And I only have half a minute to tell you that, so I'm going to skip the complexity. We can have coffee and speak about it. The only thing that you have to remember is the following, which is that one of the signature, I'm going back. One of the signatures of this interaction is that the splicing factors always recognize very specific introns. Each of them have different combination of introns they recognize. And when they are gone, mutated, and I'm finishing half a second, they actually not only prevent the normal splicing, but they actually create small versions of the transcript. And these small versions are toxic. They are making small proteins that are interfering with the normal activity of the protein. So I'm going to stay there. The puzzling thing is that we find these introns out there in the normal situation, and we believe that this interaction splicing factor to intron exists out in the axons. I'm cheating big time, oops, with the fourth slide, just to make the point that this is a team effort. The most important thing in research is that it's a team effort, and I think we should cherish it and really make it work much better than we do sometimes. That's it.